Good morning everyone. Today I am going to discuss about DNA isolation process. Okay. So as you know that DNA isolation is a process of separating the DNA from the cellular components. Okay, we will separate out the DNA from any kind of cell. That can be a plant cell, that can be animal cell, or that can be a bacteria. These are the common sources that we can use to separate out the DNA. Okay, and what is the importance of it? Why we separate out the DNA? We separate out the DNA for further processes like cloning, like PCR, and also for sequencing. Okay, I will discuss about these three processes in my next videos. Okay, so now today I am going to discuss about the DNA isolation. So for the DNA isolation, for the first step will be to break down the cell wall or the cell membrane. For the plant cells, it will be the first step will be to break down the cell wall, and for the animal cell the first step will be to break down the cell membrane. Okay, and then what will happen? We have to remove all the proteins, lipids and RNAs, that is undesired materials that we do not want. Okay, so we have to remove all those. Okay, and then we have to use this ethanol, okay, to precipitate out the DNA that we want to isolate. Okay, so these are the main three principal steps for during the DNA isolation process. Okay, so first you have to collect the sample. Obviously the sample can be a plant cell, can be an animal cell, or can be a bacteria. For different kinds of experiments the sample will be different. Okay, and then we have to use different enzymes and chemicals for the cell lysis. What is cell lysis? That is the breaking down of the cell wall or the cell membrane. That means the lysis. Okay, we will break down the cell membrane or the cell wall by using different enzymes or chemicals. Okay, then we will use some protease enzyme to remove the protein, and we will use RNase enzyme to remove the RNA, that is the undesired product, during our experiment. Okay, next is we have to precipitate out the DNA using the alcohol. Okay, so after the precipitation of the DNA by using the alcohol, we can use a buffer solution in order to wash the DNA. Okay, and then we can use it for the further study. Okay, so now we will study about how to isolate the DNA from the plant cell. Okay, as we know the plant cell have a rigid cell wall, right? So for breaking down that rigid cell wall, we need some detergents, okay, to break down the cell wall and then the cell membrane. Then we will use the RNAs to remove the contaminants, that is RNA contaminants. Okay, because we don't want any kind of contamination during our isolation of the DNA, so that is why we will use these RNAs. So the first step will be to use the detergent in order to break down the cell wall, and then we will use the RNAs in order to remove the RNA. Okay, and then we will use the chilled ethanol, okay, in order to precipitate out the DNA. Then we can easily isolate the DNA from a plant cell wall. Okay, this portion is there in your practical syllabus also, right? Next is the DNA isolation from an animal cell. Okay, in animal cells the isolation of the DNA is much more easier than a plant cell, because they do not have the cell wall, right? Animal cells do not have the cell wall, right? So that is why we will just use the lysis buffer with detergent and then protease enzyme, okay, in order to remove all the proteins that is present, that are the contaminants during the DNA isolation. So that is why we will use the protease enzyme and then the detergent. So for the animal cell we will just use the detergent and then the protease, right, in order to remove the proteins, and for the detergent is to just to remove the cell membrane. Okay, then the cell will be open, and then we will use the centrifugation process, okay, to separate out the DNA from the proteins, and then alcohol precipitation will be done in order to get the DNA sample from our animal cell. So this is how we can easily separate out the DNA from a animal cell. So why we are isolating the DNA? Obviously we can use that isolated DNA in the genetic engineering, right, for recombinant DNA technology and also for cloning. Next is for forensic analysis. Obviously in any kind of crime situation we can isolate the DNA, and we can easily identify the criminal. Okay, next is for disease diagnosis. Yes, obviously there are so many genetic diseases out there, so we can easily isolate the DNA and we can detect that if the disease is present in that DNA sample or not. Okay, so for the disease diagnosis we are isolating the DNA. And then for DNA fingerprinting, this is an important step. I will again make a video on this. Okay, and next is for PCR and sequencing. So these are the application where we can use the isolated DNA sample. Okay, so DNA isolation is a fundamental step for the biotechnology and molecular biology. Yes, obviously, because DNA isolation is very, very important and crucial for many more other steps. That is what I already told, that PCR, sequencing. So that is why it is a very crucial step. Okay, and we have to maintain the purity of the DNA. That is why we are using different types of enzymes, that is RNAs, proteases, in order to remove all the contaminants, because we don't want any kind of contamination. Contaminated DNA cannot be used for any kind of further experiments. So that is why the purity is important. And also the yield, we have to be aware about the yield, that is how much amount of DNA we want. We have to take that much of sample, okay, from the beginning. Okay, and then we can also go for the PCR step, because if we want a large amount of sample, we can easily multiply it. Okay, so these are the steps that we will follow further. So different protocols are optimized for plants, animals and microbes. Yes, obviously, as we have seen that the plants will have cell wall. So that is why we will use different techniques and procedures for the isolation of the DNA during whenever we are isolating the DNA from the plant cell. And for the animal cells, the steps will be different, and for the microbes, the steps will again be different, right? So this is the total DNA isolation procedure that you need to remember. Okay, so I hope you have understood the DNA isolation procedure. It is a very important topic in your syllabus. So learn more and stay tuned for the next upcoming videos. Thank you for watching.